Okay, so I'll talk about T-cell processing and immunogenicity predictions. And this builds very much on Sinu's prediction. Actually, I'm going to cover again some of the parts that he's already talked about. So starting with a bit of biological background, I always make the same joke when I show the slide, and I hope so there's new people in the audience. So this illustrates that I'm not a good graphic artist. This is supposed to be a mouse and a virus. I had to add the ears here because before that the mouse looked like a sperm. And I had to add the labels because otherwise nobody can see what I'm saying anyway. So if you have a virus infecting its host, it will spread, go inside of different cells in the host, multiply, and spread throughout the host. Now what happens inside of such a cell is that the virus is, as every other protein inside the cell, constantly subject to degradation. So you have the virus being cut up into individual peptide pieces, and those peptide pieces can be presented on the surface of a cell bound to MHC class 1 molecules. So if a T cell then recognizes this peptide presented by MHC class 1, and this is MHC class 1 molecules are recognized by T cells that express CD8 co-receptors, then this T cell can trigger an immune response and kill the infected cell. It can release cytokine, attracting other cells of the immune system to the site of infection, and it can proliferate and form a memory T cell response. So this is all background, obviously, for MHC class 1 T cell recognition. Now, if we are looking for what in a given virus protein, set of proteins, are the actual epitopes that are recognized like this, we must essentially understand these processes that are going on inside the cell that lead to the generation of peptides presented on the cell surface. So this peptides that you find here like this, we would call those naturally presented MHC class 1 ligands. So to understand what's going on in the cell, essentially this is the mechanism I talked about. You have proteins that are degraded by the proteasome and cleaved into peptide fragments. Most of these are cleaved further to individual amino acids reused for resynthesis of new proteins. But some of these peptides can bind to the TAP transporter, which brings them into the endoplasmatic reticulum, where they again can be trimmed primarily from the N-terminal side, and then they bind MHC class 1 molecules. And these can be transported to the cell surface, where then they can be scanned by T cells, and ultimately then you have here the antigen processing pathway where you have individual peptides within a protein that are presented on the surface to T cells. Now, when you look at these different steps involved here, they are all specific in some shape or form. These are not random processes, but the proteasome will cleave certain fragments. TAP will transport preferably certain peptides, certain lengths, and with certain C-terminal residues specifically. And MHC class 1 binding molecules have a specific preference for binding specific peptides, and that's primarily what Sinu has talked about. And the reason why we stress MHC binding so much is that this is the most highly selective step. So to be very generous, maybe 2% or so of all peptides can bind a given MHC class 1 molecule versus about 15% or so of all possible peptides, we roughly estimate, are generated through the processing pathway. And given peptides that are binding and presented on the surface of a cell, we found that about for half of them, you find an existing T cell repertoire. So really the most selective step is MHC binding. That's why we always focus on it. And yeah, so we have predictors for the other tools, and if you are on the T cell tools page, you find them under T cell processing predictions, and I'll go through these one by one. So the first ones are really a set of predictors that are mimicking what happens inside the cell. So you have here predictions that combine proteasomal cleavage predictions, TAP transport predictions, with MHC binding prediction to have like a combined score. And these were trained, these predictors were made by examining data from in vitro digests of proteins by the proteasome and in vitro assays that estimate TAP transport efficiency. So in order to identify what are essentially the sequence patterns that are associated with this. And when you look at the side, you have here for the proteasome the ability to choose between immunoproteasome and constitutive proteasome, where there's some differences in their specificity. And then you have, that's essentially all you select here. And then for tab transport, you have these two parameters that go into the prediction model, which essentially are when you read the basic paper they are coming from, they are estimating kind of the length distribution that are available for tab transport. And unless you know exactly what you're doing, you should just leave these parameters as is and run these predictions. So what you have then is you put in, just like in the MHC binding predictions, your starting sequence, you select your alleles or so that you're interested in, you click submit, and you're going to get now a table that looks very similar to what Sinu showed you before for just MHC binding predictions, but you have additional scores for proteasomal cleavage, tab transport, and they are here then combined with an MHC binding score to make a total score. So the higher the score is, the better. We, in order to make them comparable, we kind of inverted the MHC score. We do like a minus log 10 of IC50 so that a higher score is better. We just add them to the proteasome and tab score, and then we have a total score. When you look at these, you can't really see it here, but the MHC score goes from like -6 to 0 or something like that. So it has a broad range, so a high dynamic range, versus the proteasome score kind of goes from 1 to 2. So it's very, very limited. And that reflects essentially that MHC binding goes from total non-binding to a very high affinity binding, versus proteasome is much less selective. It has multiple cleavage site, and ultimately it has evolved to be able to degrade every single possible protein, so it will eventually cleave pretty much everything somehow. So now actually all this work is what I did during my PhD. So I worked on this for multiple years, which qualifies me to say this doesn't really help you, unfortunately. So I would have loved to tell you that this really solves everything, but when we test how well does these combined score work compared to the MHC binding scores by themselves, we do find that they are better at predicting eluded peptides, so peptides we get off the cell. But when we are trying to say, okay, how much better does this predict T-cell epitopes, we don't see a clear advantage there. And so one of the reasons might be that the eluded peptides that you're getting in mass spec, they tend to be the tip of the iceberg. They tend to be the most abundant, best made ligands, and that might be why we are seeing an impact there and not so much on the T-cell epitope side. More trivially, and I should have actually started with that, when you look at the binding motifs of MHC molecules, the kind of C-terminal residues they like specifically, they match with what TAB likes to transport and what the proteasome likes to cleave. And that makes sense, as an MHC molecules has evolved to bind these peptides that are available for binding. So if they had like a very, very different binding specificity than these other steps, there would be a problem in that they wouldn't find any peptides that they can actually bind efficiently. So there's a lot of overlap when you look at your top-ranked peptides based on MHC binding affinity and based on processing, and the combined score isn't really that different when you look at your overall ranking. So the other issue then is that the data we have on specifically proteasomal cleavage and specifically TAB transport are rather limited. So the other thing is, like for MHC ligand, MHC binding data, you have hundreds of thousands of data points. For the proteasomal cleavage, I think we're still stuck with like 100, 200 or so peptides that have been well studied. So that might be another issue, that we just understand one process much better because we have much better data. So ultimately, our recommendation, what we do in the lab, is we stick to MHC binding predictions and do not add these processing steps, also because they add essentially complexity. If you're the next set of tools is using not these proteasomal and TAB transport predictions here, but actually neural network-based predictions. There was the NEPCHOP algorithm that can be combined with TAB and MHC binding. There is the NetCTL algorithm, and the NetCTL pan combines this with NetMHC pan. So essentially, this is a family of very similar algorithms that are built on the NetMHC kind of family of neural networks. And the key difference here compared to what I showed you before is the use of the NetCHOP algorithm, which is a neural network trained either on the same kind of in vitro digest that I mentioned before that the other tools are generated on, or directly on C-termini that are found in eluded MHC ligands. So to some extent, this also hasn't been more, and I think it hasn't been updated for also 8 years or 10 years or something like that. So to some extent, these are historic prediction methods, and I think they're good if you want to really study proteasomal digests or so. But for epitope predictions, we currently would not use these. And I'm just going to skip through this. It's a bunch of papers here. You have all these slides if you want to look at it in more detail. What we would recommend is this MHC-NP algorithms, where actually I noticed we screwed up in that there's now 2 tools available through this. This is specifically for predicting eluded ligands, and there's a bunch of, it's an example for tools that we are hosting from a Canadian group that have asked us to host this. This was a well-performing tool in a benchmark data set, and we have now through the same interface here, we are using the NetMHC-PAN 4.0 EL algorithm. That is what Sinu showed you is the best performing T-cell epitope prediction algorithm that we are aware of, and this is essentially using an MHC binding and MHC ligand elution co-trained algorithm that is essentially predicting, it is using data from peptides that were eluded from cells, so that are naturally presented, so they are essentially incorporated in them the preference for C-terminal residues for proteasomal cleavage and tap transport, and also potentially on the N-terminal side a bit, and MHC binding affinity data. So this is the by far largest data sets that we have available for training, and they do incorporate essentially some data on the preferred processing motifs in these ligands that just comes by training on ligand elution data. So, and this is available not just here, this is also on the MHC binding page. We are, at this point, we've kind of spread and added more and more tools over time. We are actually in the process of, we are going to cut them down and have fewer interfaces. So right now, we wanted to make sure that people who are specifically interested in naturally processed and presented peptides have access to what we would consider to be the most reliable tool at this point to do these kinds of predictions. And again, they come from Morton's group here, primarily. So this is all the processing tools I showed you before. In addition to the processing itself, what we are interested in is which peptides are likely to trigger a T-cell response. So we do have an MHC class 1 immunogenicity predictor. And here, what we mean by that is we're looking at peptides that are equivalent in terms of their ability to bind MHC class 1 molecules. And then we looked for, if we look at peptides that are actually known to cause a T-cell response and compare them to peptides that are binding at the same affinity to MHC, but they do not cause a T-cell response, do we find any differences? And what we found when we essentially just asked for like a frequency of amino acid residues that are in these peptides that are facing towards the TCR, and we're comparing frequencies in the peptides that did cause an immune response and those that do not, we do see that there are certain residues that have some chemical similarities, such as tryptophan, phenylalanine, which have a long side chain sticking out in the hydrophobic, that might be making them more visible to a T cell because they're essentially like sticking into the receptor. We see less methionines, interestingly less lysines. So there is what you see here actually in this would be the average frequency of residues in the whatever human proteome or so, and then you have here how many of these do you find in immunogenic and non-immunogenic peptides that bind a similar affinity. So you kind of can imagine here you can make a score based on the amino acid composition of a peptide that is look more like an immunogenic peptide or not. So this is the tool. You enter your sequences. This masks out the residues that are responsible for MHC binding that point essentially down towards the MHC molecule, and it specifically looks for the residues that are supposedly or most likely to stick up towards the TCR. Yes, that's what I just said. When you submit this, you're getting these scores back, which essentially is just going through the peptides, looking at the residues that are looking towards the TCR, and then adding up these propensity scores. And when we validated this, and Morton just told me that recent validation actually showed that this still works, including in the most recent data sets, that you tend to have a higher score in peptides that are actually epitopes than in those that do not. So here you have non-epitopes have kind of like a random distribution, both positive and negative, and in the epitopes you have an enrichment of values above 0. But what you also see, you do have some epitopes that are perfectly good epitopes that do not have that score above 0. So if you are using, this would be a kind of selection step that you should be using if you really want to limit your set of candidates to the most highly likely epitopes. You will be losing some, but you do get an overall enrichment of epitopes if you say you only select all peptides that have an above 0 score here. So summarizing these sets of tools for class 1, the processing prediction that we have for MHC class 1 peptides, they really do work better if you want to predict naturally processed and presented ligands, but they do not, in our hands, significantly improve T-cell epitope prediction affinities. They can still be useful as additional filters, specifically if you have way too many candidates to look at in detail, and then they could provide a little more stringency to your setup. Overall, we would recommend using the NetMHC 4.0 EL scores. That's also what Sino said, which are clearly the best in predicting binding affinity, and they tend to correlate best in well, the benchmark showed that, Sino showed that they are currently our best approach of directly predicting the likelihood of a peptide to be an epitope, and consider to pair these with the immunogenicity scores. That is actually something we should be also internally to weigh these two things together, because there does seem to be an independent of processing and presentation factor of certain amino acids being more likely to be immunogenic. So that's what I have for class 1. If anybody has a mini question right now, we can do that. Otherwise, I would move on to class 2. No mini questions. Fantastic. MHC class 2, and I realize, so this is one of our problems. We switch back and forth with calling things CD4 T-cell epitopes and MHC class 2 predictions and stuff, because so CD4 and CD8 is the co-receptor on the T-cell that binds to, in the case of CD4, to MHC class 2 molecules, in the case of CD8, MHC class 1 molecules. So we tend to switch back and forth. If I've confused you so far, hopefully that helped. So CD4 T-cell epitope predictions or MHC class 2 restricted epitope predictions are specifically focusing on primarily exogenous proteins that are taken up by professional antigen-presenting cells, where these proteins are cleaved by proteases. Then they are available here for binding to MHC class 2 molecules through a actually somewhat more complicated mechanism, where they have this clip peptide in them when they are made, the MHC class 2 molecules. Then there's an exchange mechanism where new ligands are bound to the MHC class 2 molecules, and they are then presented on the surface of a cell to a CD4 T cell that has, you have the same kind of TCR engagement of MHC class 2 bound ligands. So as Sino was mentioning, MHC class 2 ligands, peptides, they can be longer. There's not a clearly defined motif. So these trimming steps that happen here of peptides before they can bind HLA-DM, they can be, they can go from very, very long fragments down to something like 13 MERS is typically when the peptides start to be too short for binding. So looking here at this specifically, we have two sets of tools, an MHC2-NP and a CD4 T-cell immunogenicity score, and this NP tool kind of mimics the processing that I talked about for class 1, and for the immunogenicity score that mimics the class 1 immunogenicity prediction score that we have. So for processing for class 2, we had looked for essentially the ends of peptides that have been identified by MHC class 2 ligand elution assays. So when we look at them, we find a very clear cleavage motif that indicates that there's specific proteases in the lysosome that are responsible for making these cuts. You see, and I don't think I have a motif here, but it's like there's there's a typical protease cleavage sites that are highly enriched at the end of these peptides. But again, like these peptides are long, and in the middle, somewhere in the middle, is going to be the 9-mer binding core that really is responsible for engagement of MHC, and the TCR primarily recognizes the residues that are bound to the MHC class 2 molecules, and there can be overhang of very highly varying lengths and all of them will be kind of having quite similar recognition properties for CD4 T cells. So when we use these algorithms, we were really happy because we were able to get much, much better at predicting the specific ligands that can be eluded from MHC class II molecules. So it works, again, you're getting much better at identifying what this, the repertoire of peptides is. However, when we then try to predict epitopes, we did not get a significant improvement, which is super annoying. And it took us a while to understand it. And one of the issues is essentially, again, the binding of a peptide to MHC class II is highly selective. And then for a given 9-mer core that binds an MHC class II molecule, you have something like 60 different peptides ranging from length 13 to 25, which is the length you typically find, that can be covering this 9 residues. So within these 60 or so possible peptides that are that contain this 9-mer, there is going to be multiple variants of there that have the cleavage motif that matches very well. So there isn't, again, if you have a good binding core, the likelihood that you'll have cleavage sites that allow you to liberate that binding core from a protein is very high. So we simulated that, and we didn't find that it was unfortunately explaining why we don't gain in the ability to predict epitopes with this. So after having told you that it's, as Morten pointed out, there have been a number of Nature papers coming out recently of people continuously falling in this trap. And the only thing that they're showing is that they get better at MHC class II ligand predictions. But none of them is actually showing any kind of improvement of T-cell epitope predictions. And in my mind, this is essentially explaining that discrepancy. Okay, so this is how the tool works. You put in your sequence in there, you do submit, you've seen this now, and then you're getting for a given peptide the N-terminal motif and the C-terminal motif. These are essentially like 3 residues on the flanking side of there, and we can come up, translate that into a cleavage probability score and a percentile rank. And again, this is now going to be much more likely to be, if it has such a good rank here, this is going to be a highly likely residue, peptide that can be liberated by these proteases. But if it's going to be an epitope or not is going to depend a lot more on the presence or absence of a 9-mer binding core that matches the class II molecules in that domain. So you can get the, here this gives us the top 5 peptides per protein. You can get the complete results. You have the probability scores. All of this is explained in this peptide. If we take just the binding or just the cleavage score, we can see like in this, we can combine the two and get a better score, again, for predicting MHC ligand, the specific ligands that are presented on class II molecules, but we do not improve really above class II, the 7 allele method that Sino presented, which just looks for presence of a good 9-mer binding course in those peptides. And here, Morten's group kind of at the same time generated a similar tool, also finding a highly a clear footprint of the antigen processing machinery, and that boosted significantly natural ligand predictions, but didn't really have a significant impact on T-cell epitopes. So that is the MHC2-NP prediction tools, and then here are the CD4 T-cell immunogenicity predictions. Thank you. I need that. So this is again an attempt of looking at, are there properties in peptides beyond MHC binding for MHC class II that can directly be used to predict immunogenicity. And this tool essentially looked for proteins from the IEDB where overlapping peptides were used from the start to the end of the proteins, and then all of these were tested for T-cell recognition, and then asked, can we train a neural network that directly identifies these based on their immunogenicity data, independent of the specific MHC alleles that are present in the donor? So are there like general properties that make peptides immunogenic? And yeah, I think I can skip over these. So then you have the same kind of setup. You have you put the protein in there. EPO is one of the examples where actually a lot of immunogenicity studies have been done. So this was a very well established data set that could be taken from the IDB for these purposes. And then we take the, here in this case, the combination of the IEDB recommended, of the 7 allele binding predictions and this immunogenicity score. And then we can submit this, and you're getting these, the median percentile rank. This is the 7 allele binding predictions and the immunogenicity score. And then there's the combined score that integrates these two. So when you look at the prediction performance, when you're combining this immunogenicity score and the MHC binding score, you do see that a combination here with a factor of 0.4, that's how the combined score was derived, gives you a higher AUC than either this must be, this is binding alone, and this is immunogenicity alone. But you also see that, again, the improvement here, you're going from an AUC value of 0.703 to 0.25. This is statistically significant, but it's not a game changer. So again, like the vast majority of prediction performance you're getting from the binding predictions by themselves. So summarizing for MHC class 2, similar to class 1, we see an enhancement of prediction efficacy for there's a minor improvement beyond MHC binding predictions in these using the immunogenicity or the naturally eluted ligand predictions, but the predictions of naturally eluted ligands themselves is greatly improved. I'm hoping I'm not too confusing here. So the epitope predictions itself, which typically epitopes is what we really care about, is not greatly improved with these methods. The prediction of the ligands that are naturally presented on a cell tends to be massively improved, specifically by the processing prediction. So the other thing that we need to point out here, I think this is very much still a work in progress. So we are constantly looking at new data sets, and we actually have standing benchmarks that where we constantly look for data that is newly made into the IEDB, and we compare different prediction approaches, how well they could have predicted the outcome of these kinds of experiments. And we're using this constantly to reevaluate what is the best and most reliable approach of predicting epitopes. And with that, I'm way faster than I thought. And, so now you can ask all the questions. Morten.
Just wanted to comment on two things. So mass-spec eluted ligand data, as Björn was saying, contains somewhere hidden signals of processing leading up to them being generated and shown on the cell surface. And incorporating them without incorporating the signal of processing explicitly, just the data themselves, the peptides in the training data set, improves performance also for epitope recognition, not only for mass-spec eluted ligand data. So what Björn was saying, I think if you try to incorporate an explicit signal of processing, you don't improve predictions for epitope recognition, but just training on these mass-spec data improves, because beyond processing, they also contain information about stability and other things, because if you have a mass-spec ligand, you have to be on the cell surface long time enough before you harvest these MHC molecules off and do your mass-spec assay on them. So they have, and the chaperones that are involved, typazine for class 1 and DM and other things for class 2 presentation, are shaping the repertoire of the peptides you find on the cell surface bound to MHC. And those you don't, these molecules are not involved in vitro binding assays. So that it seems that you gain performance by training on these new mass spec ligand data types, also for finding epitopes.
I think we need better slides with this. So Morten and I have been discussing this for a while, and I think some of the things we have been saying, we need to actually show rather than just tell. So I mean, maybe one of the things, when we do peptide synthesis for MHC class 2 for testing, So you make a prediction, you have your 9-mer peptide that has the predicted binding core. If you then make a 15-mer that has this 9-mer in the middle, or you make a 16-mer or 17-mer or an 18-mer or a 19-mer or a 20-mer, they'll all work pretty much exactly identical. When you put them in ask a T cell, can you recognize it after you pulse it on an MHC class 2 cell? Because if you give this peptide externally, it doesn't need to be cleaved. It's already liberated. It just needs to bind MHC2. And so for the vast majority of cases, it totally doesn't matter. So when we look at the T cell epitopes in the IEDB, they are not the natural ligands. They were typically directly synthesized and tested. So that the ends aren't, because they are not important for binding, because as Sinu showed you, the groove is open. So if you add some more residues on the side, you do not change the affinity of the peptide. If you do this for class 1, if you go in with the exact ligand that has the right ends, it'll bind much, much, much better because the groove is closed. You can start with a longer peptide, and it will still work because you have proteolytic cleavage of the peptides that you add in an animal or something like that, and it'll liberate ultimately also a smaller fragment. But if you start with the right fragment for class 1, it gets much better. So this is one of the things we kind of realized as we were looking at our data, so like the cleavage motifs that we find in the ligand elution data, they're simply not present when you look at T-cell epitope data, because the ends that we're using in the peptides for T-cell epitope recognition, they are not the ones that are naturally made. And knowing them doesn't really help because you always have some kind of cleavage site to allow you to liberate it. This is kind of a hard to convey message, and I'm hoping it makes sense. I'm going to quiz you all on this. Yes, please.
So if I was to design an antigen, would it be beneficial to incorporate cleavage sites, or could I just leave those out?
So if you have a natural protein sequence, I would guess you will have naturally occurring cleavage sites in there. So it is definitely, if you're designing it, why not look for, do you have good cleavage sites in there? It would be fantastic to actually try this out and very, design an optimal cleavage site. Can you get a better efficacy of your vaccine, assuming that you deliver it in a way that requires cleavage to liberate your epitopes, right? It'll depend a lot on what is your delivery system and
Okay.
There's been in the past much effort in making epitope mini genes or string of beads, and people have tried to put cleavage sites or put the natural sequences and or not, or just alanine spacers, and the data in the clinic really didn't support one way or another. So it's.
But it also didn't disprove it, and so they had in many of these contracts, you did have differences in the constructs, but it was really hard to narrow down what the reason was.
But in
that's fair or not?
I think that different people use different strategies, and the results were not very different. But
let's be
but
clear.
On the other hand, the mini genes DNA approach never worked. Period. So in that respect, it's.
Yeah. I mean, okay. So other differences. I remember still the what was it? The Kessler paper, the case relief, or so. So there is a certain advantage of having a certain length to your peptides that you immunize with. So that was a pretty clear. Okay.
I'll just jump in and add and amplify what Alessandro said, that these string of beads approaches were notably unsuccessful in terms of actually being immunogenic. Whether or not they were cleaved, there's just really very essentially no immunogenicity, even of these very good potential known epitopes strung together. They just ended up not being immunogenic.
One more. So would you say that the flanking residues for MHC2 epitopes, they're largely unimportant? That's kind of the the gist I'm getting, based on like
So
providing the-
obviously there is some kind of importance there. So in okay, when we've been using, I mean, when we're looking for, say, picking epitopes for a diagnostic or so, it is completely doesn't matter at all what the flanking residues are, because you use this as a diagnostic to recall a T-cell response, and it doesn't matter at all, pretty much, what the flanking residues are.
But they have to be there.
There have to be some flanking residues, yes. So what they are doesn't matter, but you have to have them. You must have, if you just test a 9-mer, you're screwed. It has to be, typically we say 13 residues is what you want to have. What they are specifically doesn't seem to matter, because, yeah. There were more questions also. I think there were some, yeah, Austin? Okay, so the question was, does not MHC 4.0 predict mouse alleles? And there were some specific 2 alleles mentioned, and it does, and it is, Morten should correct me if I'm wrong, and it does perform, also in our evaluations, yeah, I mean, Sinus data was mouse alleles, and it works extremely well for that purpose. Well, we would say we don't use them, just stick to the binding predictions, and that's it. I mean, using the, whatever, NetMHC, what do we have now? Is it the 2, NetMHC 2 pan? Is it still 328, or do we have 4.0? So, okay, yes. NetMHC 2 pan is also for class 2 the best predictor that we have right now, and I think we're still in the process of
One of the challenges that we face is when we design the peptides based on overlapping peptide strategy. Reviewers sometimes they pick on us saying that they may not be naturally presented. So based on what we are learning, do you think these prediction tools, whatever they predict, would be more of natural presenters?
So first of all, yes. If you do an overlapping 50-mer scan or something like that, obviously those will not. This will. It typically, again, like in our hands, it doesn't. For applications that we have in mind, it doesn't tend to matter, because we are providing the short peptides, and even if they are unnatural, as long as they contain the core of the natural epitope, they work. Yes, please.
My question is about the immunogenicity tool that you showed. What's the cut point that you would be using?
0. So it's like
everything
that's positive is enriched. That has worked for us best in the past.